In this lecture, we're going to talk about some of the processes that are really important to the whole process of cellular respiration, which happens in the mitochondria in eukaryotic cells. And so we're just going to give a quick overview here. So cellular respiration is broken into three parts. So the first part is called glycolysis. And in this part here, this is happening in the cytoplasm. And its job is to break glucose down into 2 pyruvates. And we'll talk about what those are in a moment. The next one, the second step of cellular respiration is called the Krebs cycle. And the Krebs cycle is now in the mitochondrial matrix, in the matrix of the mitochondria. So it's in the mitochondrial matrix. And the process that's happening here is we're going through a bunch of carbon, or I guess a bunch of transformations. So these are essentially transformation reactions. And from these transformation reactions, we're going to get a few things. We're going to get, first and foremost, we're going to get ATP, which is cellular energy. We're going to get NADH, and we're going to get FADH2. And these are electron donors, these two things. And again, we'll talk more about that as we go through here. And then the other thing to just keep in mind is that these first two processes are what's called anaerobic processes. And so these processes don't require oxygen. So oxygen doesn't go in here quite yet. But what does happen in these processes is glucose goes in, and out comes 6 carbon dioxides. Okay, so we're breaking this glucose apart. Remember, glucose, if you remember, is C6H12O6. And those 6 carbons from glucose get split into 6 carbons in carbon dioxide. But you can see that no oxygen has been used in those first 2 reactions, or those first 2 steps here, right? So no oxygen has gone in. Right now we're taking glucose as a reactant, and we're producing carbon dioxide. We're breaking this down into 6 carbon dioxide molecules. Okay, and then the final step of cellular respiration is called oxidative phosphorylation. And it has 2 parts that go with it. There's the electron transport chain, and there's ATP synthase. And we'll break this down a little bit more once we get into the actual process of oxidative phosphorylation. But really what we're going to get out of this process is we are going to get heaps of ATP. This process here, this last process, makes a lot of ATP. The first 2 processes make some ATP, but not a lot. Okay, so glycolysis and the Krebs cycle, they do make some ATP, but they don't make a lot of ATP. Now, opposite to glycolysis and the Krebs cycle, those are anaerobic processes. Oxidative phosphorylation is an aerobic process. It does require oxygen. It needs oxygen. And we know that because of this oxy part here, right? Oxy means oxygen. So oxidative phosphorylation is a process that uses oxygen to make a lot of energy. So here's a diagram of how this works. And so you can see here's glycolysis. And again, like I said, we've got that glucose is being converted into 2 pyruvates. These pyruvate molecules here, they're going to go into the mitochondria, and then they're going to get changed into they're going to enter essentially the Krebs cycle. There's one little step here before. We'll talk about that in a minute. But then it goes into the Krebs cycle. And as long as the cell is producing acetyl-CoA, then this Krebs cycle keeps turning. And like I mentioned, and so this is happening in the matrix of the mitochondria. You'll also notice that glycolysis is out here. It's in the cytoplasm, right? The cytosol, which we can also, they are slightly different, but we can say that the cytoplasm and the cytosol are the same thing. So glycolysis is happening in the cytoplasm. Glucose is being changed into 2-pyruvates. Those 2-pyruvates, they move into the matrix of the mitochondria where the Krebs cycle occurs. And both of these processes, we're making some ATP out of these processes here. Glycolysis makes a couple ATP and the Krebs cycle makes a couple ATP. But mostly they're making these electron donors called NADH and FADH2. And those things are moving over to the electron transport chain. So you can see that these things are being passed. These electron donors are being transported over to the electron transport chain. And the electron transport chain is in the inner membrane of the mitochondria here. Okay, and this process here is making a lot of ATP. It makes 34 ATP, much more than glycolysis and the Krebs cycle combined. Okay. In total, in optimal conditions, we're making about 38 ATP per glucose that goes in. Okay, now the key to remember here is that this is a theoretical yield. So in other words, an amount that we get in optimal conditions, which are not always met. Okay, so the cell is not always in optimal conditions, and this is a theoretical limit, not an actual limit, or not an actual yield. Okay. All right, so let's talk about glycolysis. Like we said, glycolysis occurs in the cytoplasm. It does not occur in the mitochondria. So we're not in the mitochondria quite yet. What's going in the reactant is glucose. Okay, so glucose is going in. Remember, it's a 6-carbon molecule. Really, we need to track these carbons here. Okay, so this is a 6-carbon molecule. We can count them here. 1, 2, 3, 4, 5, 6. 6-carbon glucose. And what is going to come out of this is two 3-carbon pyruvates. So we can count the carbons again. 1, 2, 3 in that pyruvate. 4, 5, 6. Another 3 in that pyruvate. So we've got 6 at the start. We've got 6 carbons in glucose, and we've got 3 carbons in each pyruvate. So there's 6 carbons still at the end. They haven't gone anywhere, right? Remember, law of conservation of mass. Now, what we get is we get some byproducts out of this. We get some byproducts, and these are very useful. We get 2 NADH per glucose. Okay, so 1 glucose goes in, and it's split into 2 pyruvate. We get 2 NADH, and we get 4 ATPs. Sorry, I was wondering about this. We get 2 ATPs are made per pyruvate, but the problem is we also have to spend an ATP. We have to spend ATPs up here. So the net, we actually create, this process makes 4 ATP, but it uses 2 ATP. So the net total, right? This is the net, these are the net totals. Actually, let's put them up here. The net totals. Okay, so the sum of everything that was used and produced is 2 NADH, and 2 ATP. These NADH, they are going to donate their electrons to the electron transport chain whereas the ATP is energy for the cell. The cell can use that ATP for energy right away. All right, now the Krebs cycle is a little bit more complex, but now that pyruvate is going to move into the mitochondria. So here we're starting with 1 pyruvate. Okay, now pyruvate is also called pyruvic acid. So pyruvate, sometimes also called pyruvic acid. And this happens also down here with this molecule here called citric acid. Citric acid is also called citrate. Okay, so this could also equal citrate. So you can see kind of both of those names are used sometimes. All right, but let's go back. So the Krebs cycle happens in the mitochondrial matrix, right? It's happening in the mitochondrial matrix. And the whole job of the Krebs cycle is to produce these electron donors. Those electron donors are going to go to the electron transport chain in the last step of aerobic cellular respiration. Okay, those electron donors are called NADH and FADH2. Both of these, they carry high energy electrons. Okay, and these high energy electrons, their high energy, can be used to do work. Like for example, so such as powering pump proteins. Right? Remember, these are for active transport, right? This is active transport. So pump proteins, we've talked about those before. Pump proteins, often they use ATP as their energy source, but sometimes they can also use high energy electrons as their energy source to power that active transport, okay? So that's just something to keep in mind before we move on to the next part of cellular respiration. And then carbon dioxide is released as a byproduct of the citric acid cycle, or sorry, the Krebs cycle. The Krebs cycle is also called the citric acid cycle. As we'll find out in a minute why that is. Okay, so if we look closely at the Krebs cycle or the citric acid cycle, what happens is pyruvate enters at the top, okay, and there's one step where pyruvate gets changed into a 2-carbon molecule here. So you can see we've got 1, 2, 3 carbons in pyruvic acid, and now we've got this molecule called acetyl-CoA that only has 2 carbons. So what happened here? Well, what actually happened is one of those carbons got removed from pyruvic acid, and it's been lost over here as carbon dioxide. Okay, so here it is over here, carbon dioxide. So anytime we go from like 3 carbons to 2 carbons, we must have lost a carbon, and usually we lose it as carbon dioxide. Okay, now this first step here of going from pyruvate to acetyl-CoA also produces an NADH. So here NAD plus is changed into NADH, and now we've got a high energy electron donor, right? So here we've got an electron carrier over here, or an electron donor that's got high energy electrons in it, okay? Now acetyl-CoA is going to enter into the Krebs cycle, and it's going to join up with this 4-carbon compound down here. It doesn't really matter what the name of it is. So the 4-carbon molecule here is going to join with the acetyl-CoA, the 2-carbon acetyl-CoA here, and they're going to form a 6-carbon, 1, 2, 3, 4, 5, 6-carbon citric acid molecule. Okay, so now we've got this is really the start of the cycle part. And then what's going to happen with that citric acid is it's going to go through a series of transformations. So we're going to go from 6 carbons, let's write this in. So here we've got, oops. Here we've got 6 carbons. And this is going to get changed into a molecule down here, which is a 5-carbon molecule. 1, 2, 3, 4, 5. 5-carbon compound, right? And so that's all we need to know is that it's a 5-carbon compound. And because we've gone from 6 carbons, to 5 carbons, that means that we must have lost a carbon dioxide. So this process here will lose a carbon dioxide. And then this 5-carbon compound is going to get converted into a 4-carbon compound, 1, 2, 3, 4. And again, because we went from 5 carbons to 4 carbons, we must have lost carbon dioxide. And there it is. We've lost a carbon dioxide. So you can see starting from pyruvic acid or pyruvate and going to acetyl-CoA, citric acid, 5-carbon, 4-carbon, and then back to citric acid. Now we've lost the 3 carbons that were originally in pyruvate. 1, 2, 3 carbons in pyruvate. We've lost them as 3 carbon dioxides. 1, 2, 3 carbon dioxides lost. In the process of the cycle part of the Krebs cycle, we get more NADH formed. So we've got more NADH here, 1. Another one down here, 2. And then up here is another one, 3. 3 more NADH are formed. And we formed an FADH2 here. And we formed an ATP right here. So we get another 8. So we've got an ATP. So in total, we formed 1, 2, 3, 4 NADH, 1 FADH2, 1 ATP. Okay, so per pyruvate, per 1 pyruvate, okay, we start with 1 pyruvate. We get 3 carbon dioxides. We get 4 NADH. We get 1 FADH2, and we get 1 ATP per pyruvate. Now, we didn't just remember that we started with 1 glucose, and 1 glucose made 2 pyruvates. So we can also consider this as a factor of, or as a function of 1 glucose. And so all we would do is we would multiply this by 2, this whole process by 2. Essentially, it's 2 turns around the Krebs cycle. And so we double the products. Okay, so in this case here, per glucose, we get 2 pyruvates per glucose, right? We're talking per glucose. We get 6 carbon dioxides are released because we had 2 pyruvates go into the Krebs cycle. So those 6 carbons in the 2 pyruvates, they get released as carbon dioxide. We get 10 electron donors. 8 of those are NADH, and 2 of those are FADH2 per glucose, and we get 2 ATPs per glucose.